So you found the skeleton. How would you tell it to represent you personally? How would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
I checked the live chat speed
right.
of sound, I think.
Oh, we are live. Sorry about that. So I am going to shrink though my little buddies here because they're taking up pretty much all the space that Walker should be occupying. So there we go.
Oh, well, hi.
All right, so I will say this: a few people are in the chat, and I know they requested to come in, and I have links going out to a few of you. I think that's probably going to be it for links. I have sent one to Otangelo. I might make an exception if you are a creationist, and either I don't know you, or I know you to be someone who is a good person to talk to, which doesn't mean I think that I agree with you. It just means that you're not a jerk, is basically what it comes down to. But yeah, this is the after show, if you haven't seen it, for the debate that I just had with Otangelo Grasso. It is not currently in the description because I'm lazy, but it will be in the description, the link to that debate if you watch this later. Also, I'm going to be mirroring it on my channel probably in about a week and a half, and that link will also end up being in here once that link is available. Oh, and Rodent No Last Name says, nice Dungeon Master screen, by the way, and thank you. Yeah, it's the 4th edition Dungeon Master screen, which is a system that I don't actually use, but I really like the art on it, so that's why I use that as my Dungeon Master screen. And it's 100% a real physical screen that actually exists, and it is not just a reproduction that I made in 3D. So, what I normally do for these is I go around in the order top to bottom, left to right, you know, and ask people for their initial thoughts about the debate. So let's start with, and this is from my perspective, they might have a different order. So let's start with Mr. Wilford.
Oh, I thought you did fantastic. It went about the same way it went about how I expected it to, because I watched Otangelo's discussion with Dr. Dan, and it was almost point for point the exact same thing.
That's what I expected.
And worse, he did a move that I will mock as like they want to say it, but they won't. He actually just said it. He basically said, I'm going to define myself into being correct, and then was stunned that nobody found that impressive.
That was my impression too, and it also, it somewhat confuses me the degree to which he doesn't realize that that's what he's doing.
Yeah.
Because he'll insist that it's still a good definition, and I'll say, okay, well, can we, because you know what, it's reasonable to say, my hypothesis has failed, and rather than completely abandon it, I will modify it. That's perfectly fine. What isn't perfectly fine is modifying it into, I can't ever be wrong, no matter what.
Yeah, he also had a pretty bad misunderstanding of like the philosophy behind falsificationism, which would have been known if he had read Karl Popper's book, and you can find it online for free, where Karl Popper expanded on that, like The Logic of Scientific Discovery, I believe is the title. He outright said in his book that falsificationism can never provide 100% conclusive disproof, because to conclusively disprove something, the data which would provide that has to be conclusively proven, which is not possible. And he was trying to frame it in a way of, if you can't basically 100% prove to me this can happen, you haven't falsified it. But no data can ever be 100% proven, period. So complete misunderstanding of philosophy, of the philosophy behind falsification on this.
Yeah, it's very also, I want to point out, so this comes up every once in a while. Yes, I do hang out with some Geth, but don't worry. These are Geth from the timeline in which the Quarians and the Geth didn't fight a war that ended with the annihilation of one species or the other, and then with one of the endings that didn't result in the annihilation of all synthetic life. So these Geth are from one of those timelines. Don't worry about it too much. People always ask about it. It's one of the first questions I always get from people who are newer to the channel is, wait, is there a Geth? Yes, there is. Yeah, no, I tend to agree with everything you just said there, Mr. Wilford. And I will say, someone in the audience, I think it was, I'm trying to find this person, Argotha, or Argotha, I'm not sure. If I mispronounced that, I'm sorry. Oh, hi, Otangelo, you're in the chat. Otangelo, I did email you the link to come in, so you do have that in your email. I think I actually ended up sending it to James too, but whatever. So you are welcome in. CRISPR-Cas is in. And I lost my train of thought. Oh, right. Argotha had said that I didn't sufficiently prove that the things I said were irreducibly complex were. I would agree that I did not probably... I didn't spend a whole lot of time doing that. But one of the reasons for that is that the things that I pointed to, with the exception of the VPU protein, are things that are frequently acknowledged as being irreducibly complex, especially things like the eye or the blood clot cascade, or the flagellum, which in fact were Behe's chosen examples. These are his sort of examples of what it means to be irreducibly complex. The R is Goth.
Behe thought those were good enough to provide to a court of law. So if you're going to use anything as an example of can irreducibly complex systems evolve, you should use the one that the creator of the argument thought was good enough to provide to a court.
Okay, so one second. Otangelo says that he didn't get the debate link, so I am going to try to send it. Maybe I just failed. Maybe I had a tech fail. But while we're figuring out why I can't technology, probably has something to do with these short arms. Walker, why don't you tell people what you thought?
Okay, I more or less thought the same thing. I mean, he said that he was going with Behe's definition. And then when you showed him systems that met Behe's definition, well, he would just say, "Oh, well, that's not irreducibly complex. That's not irreducibly complex. It evolved." And so his argument essentially became like, irreducibly complex systems can't evolve because any system that evolves is not irreducibly complex. And okay, that's just a tautology. Like you're defining yourself into something that's true by definition. Then the argument becomes, well, can you prove anything is irreducibly complex? And then when you do that, he's just going to say, okay, well, here's like this random system. Prove to me that it evolved. And then let's say you gave him a good pathway for it. Then he would just say, okay, well, that doesn't disprove irreducible complexity. That just disproves that one example. And then at the end of the debate, when you started like pressing him on like how science and falsifiability works, right? He basically just said, oh, well, you think my idea is not scientific? Well, I disagree with science.
I just want to say, Otangelo, you should be getting a link in a few moments. I just sent an email. I'm not sure why the first one didn't work. Maybe I failed at technology. Despite what people think about me, I'm actually not incredibly tech savvy. I'm only modestly tech savvy. So
there's only so much you can do with those short arms.
Exactly. Oh, and here he is. Otangelo should be joining. So what I'm going to do is, now that we've had Mr. Wilford and Walker go, I'm going to go welcome Otangelo.
Yes, hello.
Hello. So I don't know if you've seen one of my after shows, but the way I usually start out with my after shows is I first get everyone's impression, and I usually go left to right, top to bottom. But because you were one of the other interlocutors, I'm going to have you bump ahead in the line. So if you would like to, why don't you give us your impression of the show or the debate or whatever, and then we'll move on to Andrew Cumming and CRISPR-Cas and Speed of Sound.
Well, I am trying to get your point. Maybe I'm not getting it. I can be wrong about this thing. Either it evolved or it is irreducibly complex, or it can be both. It's not how I understand it. I mean, I think the definition of Behe is pretty clear. And if you want, we can read it here. And then I can clarify what I mean when I say that.
Let's do that after we get the first impressions out, because those are usually just like, a 2 to 3 minute summary of how you think it went. And then we usually dive into it. And I also want to say to Meni Karen, who said to me, Ahlan wa sahlan Dapper ya habibi. Just say shukran. Sometimes Arabic comes up, guys. It's okay. All right, so Otangelo, what in like 2 to 3 minutes, how do you think it went? And then after we get through the whole panel, we'll really dive into that question. Does that sound good to you?
Yeah, no, I think it was a productive conversation. It was polite. We were not much interrupting each other. And it's interesting to see how you guys see that whole thing about irreducible complexity, the viewpoint of yours. And I can understand why you think that the argument is not compelling, but I hope that I also brought clearly up my point and how I see it, and maybe that can be productive to have further conversations.
Okay. Well, Andrew, what do you think about how the debate went?
I think I would have to side with you, Dapper. One thing I noticed was sometimes, and this comes up, that in terms of irreducible complexity, sometimes you have to distinguish between X is impossible to evolve and X has or has not evolved. As in, like a hypothetical scenario, this is impossible to have evolved because it's irreducibly complex, whereas you can show I'm not exactly sure how to word it, but like that's kind of one point I want to make. Otherwise, I think you did good.
Thank you. Chris Burkess?
I mean, I think it went pretty well.
Okay.
There's some obvious things that maybe I would have pushed a little harder, but that's just me.
I will say I think I would have done better with more than 8 hours between issuing a debate challenge and having the debate. I'm used to having like a week or so turnaround time, and this was I messaged James, and he was like, "Can you do it tonight?" I was just like, I guess. So I will say I do wish that I had more time, and I'm sure Otangelo, you do too.
Well, I made the introduction in an hour, so
yeah. Well, then good job for 1 hour. That was actually if that was all done in 1 hour, I'm impressed. That was a pretty thorough look at the kind of the foundation of what irreducible complexity is. So.
Well, I tried principally not to bring up intelligent design literature, but actually examples where mainstream science papers or journals or magazines propose irreducible complexity, and I think I have brought up a good selection in regards of that.
Yeah. Chris Burkess, did you have anything else besides that, or should we go on to speed of sound?
No, that's about it.
Okay. Speed?
Let's see. I thought you did really well, Dappers. It is explaining falsifiability and why why irreducible complexity, the idea doesn't work. Very, very logical. I thought Otangelo missed the point on all of that and didn't come back with anything that was reasonable. And I think understanding why irreducible complexity fails is how you understand evolution. When you understand why that fails, you understand why evolution succeeds. And it's just, but there was a lot of interesting stuff, and you did a really good job of drawing Otangelo's arguments out. So kudos. Anyway, that's all.
Well, thank you. All right. So Otangelo, let's get into what you wanted to discuss, because now that we've gotten through the panel's initial reactions, that's when I like to then start digging back into any arguments.
Okay, maybe I am misunderstanding what Behe says when he writes, because any precursor to an irreducibly complex system that is missing a part is by definition non-functional.
Yeah.
An irreducibly complex biological system, if there is such a thing, would be a powerful challenge to Darwinian evolution. So that's the key issue.
There is no such thing.
I am open to correct myself if I am misunderstanding this point where he says, because any precursor of an irreducibly complex system that is missing a part is by definition non-functional. What I understand by that
there are exactly no irreducibly complex systems by that definition.
Wasn't the HIV one an example of something that didn't do really much of anything before, and it took a specific sequence of mutations to make it work?
It did something else entirely, and now it still does that thing.
Okay.
But it also has an irreducible. So you know about functional sites,
so
right?
everyone watching knows I know nothing about biology. So
let me knock out Otangelo's preferred example of photosynthesis. Okay.
Hey.
Knock out knock out anything from the photosynthetic that he wants to point at, except for chlorophyll, right? Well, only have chlorophyll. It makes stuff green. That's a function. So does it have no function? No, it has a function. It makes stuff green. That's a function. And we can build on it from there. So the idea that the photosynthetic pathway is irreducibly complex is now demonstrably wrong. He has to drop it.
Well, can I respond to that?
No, yeah.
I think what often is not taken into consideration is that the system must maintain its basic function.
That's wrong. It's why it's not.
That's the definition
it's not
of
taken into consideration is that it's wrong.
Sorry, that is the definition of irreducible complexity.
Right. Then irreducibly complex systems don't exist. You've just defined yourself out of existence.
How? How?
Because they don't exist. If you're saying, well, if it doesn't maintain the same function, then it's not irreducibly complex. There's no reason for it to maintain the same function, so irreducible complexity is nonsense. You have defined yourself out of existence.
Well, I can just give you an example. What is the basic function of a primordial first living cell? Okay, so in order for you to have that, you need a minimal number of parts.
Can we point out that this is a complete moving of the goalposts?
No.
And also a burden shift. And also a non sequitur.
Absolutely
you
not.
Completely lost the plot.
Why?
Because you're not talking about the thing you started out talking about. Look, are you saying that photosynthesis is irreducibly complex?
No. I want
an answer to this question. This is important. Are you saying that photosynthesis? You're saying that it is.
Okay. What is the function of photosynthesis?
It doesn't matter.
Of course it matters.
Because if you take a part out, it makes stuff green.
Wow. So you don't understand the argument.
Are you saying it does make stuff green?
You don't understand the argument.
Hold on.
We're getting a lot of crosstalk. Okay. Otangelo, why do you think that it is vital what the particular function is in order to declare something irreducibly complex?
Okay, because look, by irreducibly complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function. So let's just stop here now. So what is the basic function of photosynthesis? It is to produce glucose.
Okay.
Is it composed of several well-matched interacting parts? Yes.
Okay, CRISPR? Now explain why you think that function, what it is in particular, is unimportant, or also I just want to point out we have a $2 super chat from Sphincter of Doom. Hi, Josh. Who says chlorophyll also facilitates charge separation. I did not know that, but look, I'm going to point out
in the reaction center. Yes,
actually a great example.
I am not biochemistry great at, so I'm more about the bones. So I just wanted to point that out. But yeah, go ahead, CRISPR.
Well, it's a fascinating thing he points out about charge separation is that you have one possible function, right, of making stuff green, which results in an ancillary function.
That's not
of increase.
Function.
Hold on,
I'm not done yet. Of increasing the rate of charge separation, which then allows for further functionality of a metabolic process that was not possible before, right? So this is a great example of exactly the way you can build on unrelated functionality to produce unrelated functionality. Now, you ask the question, and it's a great question, why is it that it doesn't matter what the basic function of a modern system is for whether or not it can evolve? And the reason is that evolution doesn't care about what happens in the future. It cares about what's happening right now. Okay? And so if right now you have a system that does something useful, right? And then in the next generation you have a minor change, and now you can do something completely different with it, fine. Doesn't matter. You can still build on it from there. And so what he's saying is, it doesn't matter if you can build the thing through a discrete series of minor modifications if the end result is doing something completely different from what you started with, even though every single step and every single stage was individually and independently adaptive. Well, that's nonsense.
All right, so can we, Otangelo, would you like to respond there?
Well, I think I made it very clear in our conversation, Dapper. I gave the example of chlorophyll, and said it requires 17 proteins and the biosynthesis pathway to have chlorophyll. And the last 8 proteins, they are not used in any other system. And if you remove any of those proteins, you will not have the end product. So that makes the biosynthesis pathway of chlorophyll irreducibly complex. Now, let's suppose that you even produce chlorophyll. So what? If you do not have all the other parts, if it is not embedded in the light harvesting complex, it cannot function at all. And being
wrong.
Green, that is not the function. That
No, I
is
mean, you're already wrong.
Okay,
You can stop talking now because you're already wrong.
So, CRISPR, why don't you explain what's wrong?
So again, chlorophyll on its own can result in increased levels of charge separation. Okay, that
In the
is
photosynthesis
Hold on,
system.
Otangelo, hold on.
that is the necessary function it performs in photosynthesis. The thing that it does in photosynthesis that's important is it facilitates charge separation.
So can charge separation be useful on its own without the actual production
Yeah.
of energy? Can you explain, like, an example? Just to make sure that we all...
So firstly, you say without the production of energy, but so that's just the sort of a non sequitur.
Oh yeah, because charge separation would produce an energy gradient.
Right.
So I guess I mean without the production of
Right.
glucose.
Well, right. So photosynthesis. So the point of photosynthesis is not to produce glucose. That's actually a separate thing, right? The point of photosynthesis is actually driving ATP synthase through its own production of gradients, right? So it's only you have to produce glucose so that the plants can then go through respiration and break down that glucose at night. That's the important thing. But that again, that's missing the point, right? So that gets into carbon fixation and all these other things. But again, not really the critical point. The critical point is, right, chlorophyll, charge separation, the critical role of chlorophyll in photosynthesis. So is just making stuff green enough to get to photosynthesis? Yes, because the thing that makes it green also does charge separation, which is the role it provides in photosynthesis. This is the problem with having somebody who doesn't understand the biochemistry try and talk about things being irreducibly complex, because you don't understand the connection between things.
Otangelo?
Well, the end function of photosynthesis is not to make ATP. It is to make glucose. That is
that's wrong.
That's the entire pathway.
Hold on. Let's let him get a little bit more out. Otangelo, could you repeat what you were saying?
ATP is made to then making NDP-AP, and from there you go to drive the Calvin cycle and to fix carbon dioxide, and in the end to make glucose. So the entire pathway.
What's the glucose used for?
Well, as energy for the entire organism.
Okay, so if it has light constantly, does it need to make glucose?
No.
Right, so it doesn't need the Calvin cycle.
No.
Right, so the point of photosynthesis is to make ATP. Thank you, that was my point.
No. The end function of photosynthesis is to make glucose.
You're wrong. You're wrong. The end product of the Calvin cycle is to make glucose.
But the Calvin cycle is part of photosynthesis.
No, it's not. The Calvin cycle is driven by the ATP produced by photosynthesis, but that is not the same thing. Again, you don't even understand how these things are connected to each other.
All right, I want to say real quick, we have a $5 super chat from Rodent No Last Name, who says, not following the prior precursor structure that allows for functional structure that appears irreducibly complex, where the precursor is no longer present, is not irreducibly complex. And if I'm understanding what he said correctly, and I might not be because my brain is a little fried at this point, sorry guys, then I believe I agree where he's saying that just because we don't know what prior system might have preceded a currently irreducibly complex structure, sorry, doesn't mean that suddenly there's this irreducibly complex problem. And I would like to talk about that a bit later, but I would like to also continue this photosynthesis thing, because I will admit, I do not know a whole lot about photosynthesis, but it also does seem to me that the fact that two biochemical cycles interact doesn't make them the same thing. And the cycle that is actually taking sunlight and using it to drive the production of ATP is not the same cycle that is then taking that ATP and using it to produce glucose. Those do seem to be separate cycles, and I'm not an expert, but it does seem to me like, Otangelo, that you can't simply say that they're both the same thing.
I've not said that. I said that making glucose is an integral part of the system of photosynthesis. Photosynthesis is not characterized only of making ATP.
What would be your definition? Oh, sorry.
I mean, you can just look at Wikipedia and there you have it. And it is an integrated process. In the end, the function is to make glucose. That's the function of photosynthesis.
Maybe we could get a definition of photosynthesis from both Otangelo and CRISPR, because I feel like maybe that might help clear up.
I can read it here if you want.
I mean, you give whatever definition. If it's one that you're getting from a source, that's fine.
Well, I mean, if you are okay, I can read from Wikipedia.
If the Wikipedia source is one you're going to stand by, I'm fine with that.
Well, photosynthesis is a process used by plants and other organisms to convert light energy into chemical energy that can later be released to fuel the organism's activities. This chemical energy is stored in carbohydrate molecules such as sugars, which are synthesized from carbon dioxide and water, hence the name photosynthesis, from the Greek light and synthesis, putting together. In most cases, oxygen is also released as a waste product.
Okay, Chris. CRISPR. What would you use as a definition?
So photosynthesis is just producing energy, energy usable by the system from light. And in all life, the form of energy that is used is ATP, right? So the purpose of glucose production, right, is a stable long-term storage of that produced energy so that it can then be used later on through existing respiratory pathways. But what's the point of photosynthesis itself? It is energy production purely, not the storage, the production.
Okay, so Otangelo, would you be willing, for the sake of just discussion, to argue about photosynthesis just until the point that we get ATP?
Sure.
Okay.
No problem.
Then I think as long as we can agree to that as sort of our boundary, I think we can then usefully continue on. So would you say that up to the point of ATP production, photosynthesis is irreducibly complex, and let's limit it to photosynthesis in plants, because do we want to go into the purple bacteria that use retinol? I don't know. That's a completely different chemical system. I don't want to get into that. Or like red green, or like red algae. I don't want to get into that either. We'll just stick with plants. Does that work for everyone?
Well, the point is that we have to say it is oxygenic photosynthesis and not with plants, purple bacteria, because you can reduce it, of course, and you don't use the oxygen.
That would count for plants. So can we agree to stick to just plants for this discussion?
Yeah, sure.
CRISPR?
Yeah, that's fine.
Okay. Yeah, I just want to make sure that we have some boundaries that we can all agree to as to what we're talking about, because otherwise we can get lost in the weeds. Okay. So, CRISPR, if I'm getting you right, your claim is that because there are simpler functions that you can perform with parts of what is now used for photosynthesis in plants, it is not irreducibly complex.
I mean, that seems to be meeting the definition of what irreducible complexity is all about.
Yeah. Okay. So
note that I say what it's all about and not what they necessarily claim it to be. Right? It's like what is it that they're driving at with irreducible complexity?
Right.
What they're driving at is it can't evolve. But if it has some prior function, it can evolve, even if that prior function is not the current function. And that's my point.
Okay. Otangelo, do you have a comment, disagreement, criticism, question?
Well, he has many times answered to this. That's the co-option argument, that a smaller part of the system was co-opted and had another function. But he says even if that is the case, and even if a smaller part of the system had another function, that doesn't mean that system is not irreducibly complex, because we are talking about different uses here. A smaller part, I don't know what you are referring to, would have another function, but it would not make ATP.
Okay, so let me ask you something. Let's say that protein A has function X on its own. Function X is adaptive, and let's say that that protein B, a modification of protein B1, is maybe non-functional, but protein B1 has function Y, right? And so basically you break protein B1 into protein B, and now it can interact with A and do function Z. Okay? Can that evolve?
But first you have to have these proteins.
But you hear what I say, right? Protein A, functional on its own, adaptive on its own. Protein B1, functional and adaptive on its own, right? Protein B is the derivative of protein B1 that is non-functional on its own, but functional in the presence and by interaction with protein A.
It's not functional on its own?
That's what I said, yes.
And why would evolution produce a protein which has no function?
Happens all the time.
It produced B1. B1's functional.
Yeah, and proteins. So a common thing that happens in organisms is that you get a whole gene reduplication, and then subsequently a mutation that breaks the protein. But because the gene still has a promoter, it still gets transcribed and turned into a protein that just doesn't do anything. That happens all the time. Your body is probably right now making at least dozens of proteins and they're just not doing anything. But the broken version can subsequently mutate to get a new function. That's how flounders in the Antarctic got their antifreeze. They had a protein.
That would be devolution.
No, you're gaining a new function because remember, so you have a whole
that
gene.
That would be evolution by entropy.
Let me finish. You have a duplication of a protein gene. Now you have two. One mutates. You still have the original protein hanging out, doing its original function just fine. Now, the old one, it might be broken right now, but then later, if another mutation happens, what was originally just a copy of a protein gene now produces a new protein that actually has a function. The old function remains intact. Now you have a brand new function. And what CRISPR is asking is, couldn't it be the case that this brand new protein could interact with two different proteins and form a newly irreducibly complex system involving three proteins? Why is that impossible?
I don't know. I would have to study this case.
No, CRISPR, I did reasonably well.
Well, I was actually my system only had two proteins.
Oh, sorry.
But yeah, it's the right idea.
Okay, yeah. So I was hoping that I roughly got.
I was trying to make it as simple as possible.
Yeah.
Yeah. So this is the issue: is that if you can have functionality that is adaptive, right, at every necessary stage, then the fact that at some point there is a change in functionality, right, doesn't matter. It's fine because it's still adaptive and it's still driving things forward. And what I'm hearing from Otangelo is, well, if it's not the same function at every step of the road, well, then it can't happen. But that's just not true.
Okay, I get your point. And if I have a paper or something, I'll be happy to read it and concede your point.
But there are already papers about this. This is one of the things that's frustrating about me. It's frustrating me about you.
I'm asking you to give me a paper, which I have not read yet. I will read it carefully, and then I will concede your point.
Well, let me ask you this.
Hold up a second. Everyone, hold up a second. I need to get this out there. Okay, so, Otangelo, I don't know if you know this, but I was a young Earth creationist for quite a while. I don't know if you know that, but it's the case. However, when I decided to investigate it, I purposefully went out of my way to make sure that I found things that disagreed with me to check to see if there was any merit to them. Because I had already read, I'm not going to say all of the creationist books, but I'd read many creationist books that were out at the time. You know, I've read some of the biggest influential works at the beginning of the modern young earth creationist movement, like the Genesis, Flood, Scientific Creationism, both versions of evolutions. The fossils still say no. I was reading articles on creationist like publications, and the internet wasn't as big at the time, but I was still going to creationist websites. But I was also seeking out things that disagree with me just to check. And what I'm hearing from you, Otangelo, is that there are things that people on the other side of the argument are saying, these are things that there are dozens of papers about. This is not even a topic that's in dispute. And it's frustrating that
it's too general to find a paper.
Well, first, there's just one paper. I mean, there are dozens of papers that all just take this for granted because it's not an open question anymore. This isn't even... I'm going to be honest. There is literally no real debate on this topic that exists in the scientific literature because there's just no there. And the fact that you haven't read any of the papers that disagree with you, which is essentially all of them, that touch on this topic, is frustrating to an extreme degree because it means that you don't care to test your idea. Or at least, I apologize, I'm sorry. That was inappropriate. It seems like that's what's going on.
I just asked you to send me the papers. I read science papers.
And I'm going to be sending you some papers here. I mean, I haven't yet.
I don't even know what you are talking about. So that's what I'm asking you. Send me the papers and I will read them. And I will be more than happy to grant your point if you have one.
I'll accept that, but it's still very frustrating.
It's a concept known as exaptation, right? Where something with an ancestral function is exapted into a new function. It was popularized by Gould. So this has been
the co-option. Argument is not
hold up, Otangelo.
No, it's not. It's not co-option. It's exaptation. That's what I'm saying. Right, so something has a new function, and then some sort of pressure comes along when you're talking about like macroscopic traits, or like a new protein modification will come along that creates a new pathway from a pre-existing protein, right? That served a different function in a different context. It's a fairly popular mechanism of evolution. Yeah, that was that's all I was going to say. It's just called exaptation. If you want to read up on it, yeah.
What frustrates me is the idea of how would you find a paper to give to Otangelo that would explain this? I mean, it's so general what we're talking about. It's so from the top. How could you find a paper about that particular subject?
The only thing I could think of is papers that touch on individual instances of things that could be.
There's also essays about this kind of stuff, and like a lot of intro courses will cover this as a mechanism. Like, you know, have it in my notes and stuff. I don't know if I could find just like a paper on the mechanism.
Yeah, I mean,
you could find a textbook article maybe on it or something in a real.
Textbooks would be a good place. Yeah.
Well, actually, that's a good idea. Otangelo, have you ever bothered with a fairly standard evolutionary biology textbook to just read through it? You have?
Yes.
Like,
have you read it?
Like, could you, like, which one?
Oh, I have an entire library, and I read evolutionary literature on a daily basis.
Which textbook is for like genetics or evolution?
Oh well, do you want me to list those which I have here in my
no,
library? I can do that.
Just your favorite. Just your favorite. I don't need a list. Like. What do you like?
Well, I like more biochemistry. Bruce Albert's That's
good. That's good enough. Thank you.
I want to say for $5 from El Spaghetto that he says, or I'm sorry, I'm just going with the gender on the word. I don't know. So El Spaghetto, if your name is masculine but you are in fact someone who uses she, I apologize, but. I'm just going to go with it. He says, imagine finding a paper that would convince Otangelo. Also, best wishes to y'all. And thank you very much. Best wishes to you. And also everyone else who's in the chat. I know there's a lot of people who are not normally on my channel. I really appreciate it. And if you would like to get more of my content, I do most weeks have 3 videos that come out, and other weeks are usually 2. So you are getting pretty reliably, and this has been the case for over a year now, that I'm getting 2 to 3 videos every week. So usually at least 1 live stream and 1 scripted video is what you can expect. Sometimes it's 2 of one or 2 of the other, but yeah. So do feel free to subscribe. Also, if you want a link to the original video that this is talking about, the original debate, if my mods could put that in the chat every once in a while, that would be greatly appreciated. And also, I'm seeing that like some 20 of you haven't hit like yet, so help me out, guys.
I haven't.
Uh-oh, you're on the panel, Mr. Wilford. Come on.
Damn, I'm so sorry.
Oh man.
Could I ask about a particular question?
I don't have a problem there.
Okay. In the after show or the question and answer period, a question was asked: how do you know that the protein did not have another function? And Otangelo, you quickly, I think you kind of blew it off by saying you look it up, it'll tell you what function it has. Well, that is the function that we know about now. Now, it's possible that it has another function that we don't know about. It's possible that it had another function in the past that we don't know about, and it's possible that it's mutated, and prior to that, it had another function we don't know about. So, how would looking it up answer that question for him? I don't understand how you just dismiss this question so offhandedly.
The thing is that we are not talking about just 1 or 2 or 3 proteins which eventually mutated to get another function. We are talking about entire metabolic networks which are required to make the basic building blocks of life. If we are talking here about chlorophyll, you need 17 proteins which are working in an ordered sequential fashion where
Okay,
one product is handed over to the next one.
Hold up, that's not the question that's being asked. Otangelo, maybe I can rephrase it. So,
Okay.
There are, in fact, lists of known functions of given proteins, right? But we don't have a list of the unknown functions of proteins, correct?
Okay.
Because how could we? That would be impossible. So how do you know that there is not a function that is unknown to any given protein that has a known function?
That's basically an evolution of the gaps. We don't know the function which eventually it had prior to the function that it has now. So we are just supposing or hypothesizing that it had another function previously.
No, we're not even hypothesizing. We're just not excluding the possibility. There's a difference between assuming X and not excluding X. There's a big difference.
I tried actually to answer precisely to why I'm not seeing this as a convincing argument, but you interrupted me.
Okay, I'll tell you what. I do want you to be able to finish, but I just want to please. It's one of my pet peeves when people misconstrue arguments that are fairly clear. The question is not how do you know that X doesn't exist. Sorry, the question isn't whether or not we should, assume X being that the protein has a different function. The question is, how can we exclude it? There's a difference. Evolution of the gaps would be assume evolution happens no matter what.
Why should you include it in the first place if you do not have any evidence that was the case?
I'm not asking you to include it. I'm asking you, how can we exclude the possibility that a protein has an unknown function? How would we do that?
Well, you are working with a hypothesis which you have not instantiated. You're just making it up. Oh, it had no function and then it got a function.
No.
But how do you know
I'm
this?
not making that hypothesis. Go ahead, CRISPR. Go ahead.
There's an important thing here, which is that Otangelo, necessary in your argument is the impossibility of any possible mode of evolution, right? Your claim requires that for at least one system, at least one, it is completely impossible for that system to evolve, right? Yeah. Which means that for at least one system, you must conclusively demonstrate the impossibility of any possible mode of evolution which you cannot do.
I can.
Yeah.
How?
No, you
I
can't.
can.
No,
Of course I can.
maybe he can. How?
Then you have to prove
Christopher-
every possible imaginable way is
Christopher,
ruled out.
I want Otangelo to explain how, because I think if he, let's say he can, that would be fascinating.
Brilliant.
It would be. He's now taken on the burden that he just said he didn't have, but I'm curious as to what he's going to say.
Otangelo, I'm actually, unfortunately, going to have to step away for just a bit. So you guys are going to have to report back to me, and I'll try not to comment too heavily on this, but I will be right back, and I trust Otangelo and the rest of you to both behave and not leave me with large amounts of dead air while I'm gone.
Oh boy.
Yeah, it's a big responsibility.
Everyone starts swearing right now.
I will watch the VOD Walker, and if you got me demonetized, you know my rule. Okay, maybe you guys don't know my rule. And if you, here's my rule. We get one F-bomb per stream. Anyone who does one after that owes me a $10 super chat, and if you don't, you're gone. So if you don't consent to this rule,
before I joined.
Well, if you don't consent to the rule, get out now.
I can afford that.
Okay, I'll be right back.
Dead air.
You probably should talk about.
Otangelo, there's one other thing that I want to bring up, and again, I'm not a biology guy, so I'll try to keep it short. But in the debate, didn't you say that the smallest known protein needs to have like 120 amino acids?
That's not true.
I can give you the one which I have.
Okay, because I'm pretty sure you said the smallest one needs 120 amino acids.
It is a ribozyme. Let me just find it out here.
Okay, well, the point is that it's you did say in the debate, maybe you misspoke, and that's fine, but you did say that the smallest one requires 120, and that's not true. The smallest one actually has 11.
Oh, 11? I thought it was 12.
So I will just tell you which one I was referencing to. Okay?
Yeah, the smallest known protein is called like the TAL protein. I just found it. It's only got 11.
Sorry, I was referencing to ribonuclease, the simplest RNA protein, and it has 124 amino acids.
Okay, well this isn't an amino acid, or this isn't an RNA protein, but this is still a protein.
What? You mean?
Or sorry?
Did you say ribozyme?
Well, it's a RNase, adenase or ribonuclease. The simplest RNA proteins are ribonuclease, and it has 124 amino acids and 19 or 20 different ones that are considered essential.
Ribonuclease. You're trying to say ribonuclease, right?
Yes,
RNase?
Yes.
Yeah.
Real
Okay,
quick.
So that's
got
not a simple protein.
We got a $10 super
It's
chat.
not the only protein. It's not anything else.
I just want to say we had a $10 super chat from El Spaghetto who just said forgo. So there you go. Our one F-bomb is apparently used up.
No, I think that means that someone on here gets 2.
Well, oh, hold up. It's not 1 per stream per person. It's 1 per stream.
Really? Oh,
yeah.
hell.
See, don't.
Yeah,
I'm just assuming I'm fast enough that it would be me.
I keep a fairly strictly PG-13 channel. So one of my goals is I don't want anyone who watches this to brush off the things that I say because of the way that they're said.
No, I adapt to whatever room I'm in. It's fine.
I have no such problem.
Yeah, well, that's fair. But also, my nieces watch this.
Oh.
So, and I told their mom that there wasn't anything that wasn't like I told their mom that nothing was inappropriate for kids, but I don't know that they would find it interesting, and I have to live up to that promise to her.
Nice guilt bomb.
Yeah.
Well, you do have the problem of your side chat. That might be a little bit.
I don't think my 4-year-old niece is reading the side chat.
Oh, well, yeah, probably not.
Okay.
So I think that Otangelo is going to undertake his burden of conclusively demonstrating the absolute impossibility of any conceivable mode of evolution for at least one system.
Yep. I'm dying.
Go for it, Otangelo.
Well, as I said in the debate, that the concept of irreducible complexity is not just constrained or restricted to evolution, but also to the origin of life. And that is in regards of the cell, which I regard as irreducibly complex because a minimal number of parts are required in order to kickstart life. And there are several science papers out there which try to elaborate what that minimal cell would be. And there is a paper from Nature from 2016. They boil it down to about 570 proteins. They specific. They enumerate each of those and they explain why it is necessary. So that is, in my understanding, something which cannot be produced by evolution, and we have
your understanding's wrong.
Well, can I finish?
What you just said.
Yeah, let's let Otangelo finish at least a couple more sentences, and then we'll jump in with whatever response.
I mean, I don't even see that evolution would be a capable mechanism because we require DNA replication in order to have evolution. So if we say chemical evolution, I mean, if we are saying chemical evolution, okay, you can go for that. But science has demonstrated in 70 years that they aren't even able to come up with a route for the prebiotic, non-enzymatic production or synthesis of the basic building blocks of life.
Okay, so let's all know. So let's all note that what he did was he was asked to provide conclusive demonstration that all possible modes of evolution are impossible, and what he did instead was say, "I don't know how it could have happened," and that's not good enough.
He also, instead of coming up with one system that was irreducibly complex and impossible to evolve, he came up with the entire cell.
It's fine. No, honestly, that doesn't bother me. That doesn't bother me that he picked the entire cell. What bothers me is he picked the entire cell and then did not demonstrate his burden.
Okay.
Support your burden.
Well, of course I can do that. Let's suppose that the numbers which I come up with, which is a minimal cell, which is, for example, a bacteria, Pelagibacter B.
No bacterium is going to count as your minimal cell. All bacteria have, according to mainstream science, billions of years of evolution behind them.
I would like
it doesn't matter. It doesn't matter because
we can take Luca, which is proposed by Nature magazine in 2016.
Otangelo.
It has.
Otangelo. I'm sorry. Here's the thing.
Guys,
here's
I'm not able to finish any sentence.
All right, let's let him finish the sentence about Luca. And
if I'm not finishing next sentence, then I will leave.
All right, Otangelo, please finish your sentence about Luca. That's fair. Mid-sentence interruptions can... Yeah, okay. Finish your sentence about Luca at the very least, and then let's take a quick pause. And then I will say something, and then we'll perhaps move on to CRISPR. So, Otangelo, finish up at least the sentence about Luca. If you have another follow-on, you can finish that one too.
Okay. If we take that example, which has been provided by Nature in 2016, it has 570 proteins, which they consider as absolutely essential to maintain the basic functions of life. Okay, so if you make the calculation, if that system would have emerged by unguided events, then you have a calculation of 1 to 10 to the 350,000. That's in the realm of the absolutely impossible.
Okay. The first thing I just want to say real quick is using Luca as a stand-in for the first living organism on Earth is crazy beyond belief. Luca was much, much after the first organism by most accounts of abiogenesis. That's all. That was my piece there. CRISPR, go ahead.
Well, I mean, he still hasn't proven anything that can't evolve. He's just said he doesn't understand how it could, and that's not a substantiation of his burden.
Yeah, that
failing his burden. He needs to support it. That's it.
I would definitely agree that. So this is an argument, and I like this term from R.J. Downard, where basically a creationist will point to something, point out how complex it is, and say, "Gee whiz, I guess God." That isn't an argument, though.
Yep.
So what is an argument? How do you recognize design?
Well, so I recognize design when I, one, know that there is in fact a way for a designer to make something. So I have a known mechanism by which it can occur. And I then don't have a known natural mechanism. So for instance, if I look at, say, let me give a quick example. So let's say I want to look at some of the most simple technology that humans have ever used, which would be just simple knapped flint blades. So I know that nature can make sharp things, but it tends not to carefully knap flint into particular ways where it becomes a useful blade that can be used to do things like separate meat from bone or something like that. However, I do know how humans, or depending on how broad we want to use with the word human, near human organisms can, and still do, because you can still find people flint knapping today, make such things. So because I have one, known mechanism for designers, and B, no known mechanism for non-designers, I recognize design. With evolution, I have no known mechanism for designer, but I do have a known mechanism for no designer. Therefore, I conclude, until demonstrated otherwise, no designer, at least in evolution. Now, I don't have a known mechanism for abiogenesis, so I remain agnostic on the point. Go ahead, Otangelo. Sorry.
Okay. When you see a blueprint and then a machine or a factory made upon the precise instructions of that blueprint, and this is made for a specific purpose, and you have two options to consider. One is that random chance made the blueprint and the machines upon that blueprint, and the second option is that intelligence made the blueprint and then the machines and factories based on the instructions of that blueprint. Which of the two propositions are more likely to be true, and which one have we observed actually to be able to make both?
Okay, but one big problem is that calling DNA a blueprint is extremely troubling. DNA is not a blueprint. DNA is more like a recipe. It's not even really much like a recipe. So, for instance, DNA, a blueprint is a picture that represents in fairly good detail what the final product is.
It
DNA does not do that at all.
Okay, DNA and epigenetic information, both, they work together and they provide assembly instructions to make proteins, and they have also regulatory functions which direct the operation of cells.
Not entirely, because epigenetics doesn't contain any information on how to make a protein. It simply allows some proteins to be made and others not to be.
So let me ask in a broader sense, is the making of the cell and proteins directed by information?
Depends on what you mean by information.
Instructional complex assembly information.
Well, you've used the word information in your definition of information. So, again, what do you mean by information?
Instructional complex information. It instructs how to make proteins and how to operate the workings of a cell.
Right. So, one of the basic rules for giving definitions is you cannot use the word you're defining in the definition. So, you need to be able to define the word information, and in
it instructs the making of proteins, and it directs the operation of cells.
So wait, all information makes proteins?
No, but a part of it.
Then that's not a definition of information.
Yeah. So
I said it instructs the assembly and making of proteins, and it has also regulatory functions and operates how the cell has to work.
Okay, so anything that doesn't do that isn't information.
Well, I am asking, are genes storing the information to operate in the way as I just described?
In order to answer your question, I need to know what you would call information in the generalist way, so that I can look at anything in the world, and if I know enough about it, I can say, yes, this is information, or no, it's not. You have not given me that, unless you would say that the only thing that can be information is something that makes proteins and cells.
Okay. The nucleotides in the genome, the sequence of them, and the genetic code, they direct the making of proteins, and they also instruct the operation of cells.
So all information consists of nucleotides.
Yeah. So my computer has no information on it.
Not only the nucleotides, but also the epigenetic
Okay,
codes.
Ask me to define information.
Hold on. First,
Between regulatory network.
a couple things. One is Speed of Sound of Gravity just gave me a $10 super chat, and he says saving up. So, oh boy. You know what? To my nieces, if you're watching, I apologize in advance to both you and your mom.
I'm paying in advance. That doesn't mean I'm going to do anything.
Fair enough. Okay. So, Otangelo, let me give you an example of a definition, right? So let's say you wanted me to define a book. I would say a book is an object containing sheets of some material on which images or written characters are applied via some relatively permanent technique, and which is bound by a cover such that one can open the cover and reveal the contents of the page in sequence, right? Now you have enough to look at any object in the entire universe, and according to my definition, you can tell me if it's a book or not, right?
Although an audiobook wouldn't be a book by that definition.
Okay.
Which one? I wouldn't call an audiobook a book, actually.
Okay, to agree with that definition, the biological significance of DNA lies in the role it plays as a carrier of information, especially across generations of reproducing organisms, and within cells as a coded repository of system specification and stability.
That's great. And Otangelo, I want to say one thing. I am actually open to the idea that DNA contains information. Like, I don't reject that out of hand. But in order to answer the question in any given instance, I need to know exactly what the definition of information is. Now, I would like to hear CRISPR's definition.
I mean, I'll say for myself, I absolutely think that DNA is encoded with information. It clearly is. Information is that which can distinguish one thing from another, right? So
I tend to agree.
So I mean, it's pretty simple to say that the ordering of different nucleotides can be considered as information by that definition. So there we go. I mean, I don't see why it needs to be any more complicated than that. But you'll notice I was asked for it. I wasn't even asked for it, but I gave a definition for information that didn't use the word information in it. And it seems like that definition will cover everything that is information. So that's how you give a definition.
Yeah, so I am willing, if Otangelo is willing to accept that definition, I'm willing to say that in that case, yes, DNA contains information. Otangelo, would you be willing to accept that definition?
Yes, sure.
Okay, then yeah, I think everyone here is. I'll say this: if anyone here on the panel says that DNA does not carry information by that definition, please speak now or forever hold your peace. I'm going to go ahead and say we all accept it then. So why is that an important aspect of DNA or biology, Otangelo?
The relationship between information directing the making of machines, factories, computers, turbines, energy plants, transistors, and so forth, and the making of these devices, both the information and the making of those things based on that information is always sourced and tracked back to an intelligent source.
But that's
I'd like to see you substantiate that claim.
Yeah, I disagree because biology is a source of information that does not come from an intelligent source.
Actually, hold up real quick.
Well, that's an unwarranted claim. Now you asked me to back up what I am saying. So
it's a non-argument.
Wow. So I cannot finish my sentence. I am tired. It's already 2 in the morning, and I am losing really my patience because you guys are interrupting me, and I am trying to make a point, but I cannot because I'm being interrupted.
I just want to say, Otangelo, I am trying to minimize that. At least acknowledge that. Like I was just about to say, let's let Otangelo finish his thing. So I am trying, although let's be fair, there are a lot of people here, so it's not easy. So I am trying, and if you'll notice, numerous times I have told people to hold up so that you can get your thoughts out. So please at least realize that I'm working on that. So Otangelo, please get your thought out.
Yeah, it will not take long. There are several science papers, Kunin and so forth, of an entire site, my website, where it says that the origin of life has nothing to do with evolution. So the mechanism of evolution does not count to explain the origin of life. When you do not have evolution, then the only left mechanism is unguided random events, chaotic events on a prebiotic Earth. So if you are excluding intelligence as the origin of biological information and the making of molecular machines and cell factories, then the only alternative mechanism is chance. And you can calculate what the odds would be to have a minimal proteome of about 570 necessary proteins to have a first self-replicating cell, and the odds, they are beyond what is possible. And I gave a number 1 to 10 to the 350,000. And even to have just one protein which would be functional, it would be beyond what is reasonably possible.
Okay.
The odds of one protein that is functional is 1 in 1.
Hold up, CRISPR. 1 second. I have a quick question, and I just need a yes or no on this one. When you gave that number, are you basing it on Luca?
I based it on the science paper. Well, you tell yourself, what do you think would be the smallest possible living
Because I don't know.
device?
I don't know.
Okay, it's a number. I mean, they came up with 560 or 570 proteins. Let's suppose even it would be 100, even 100 proteins would be not reasonably possible.
Okay, but Otangelo, I want to know, was that for Luca?
Yes.
Okay, that was the answer I was looking for. I think everyone here knows why. Walker, you had a question, and now that Otangelo, do you think you've gotten your full thought out?
Yes.
Okay, now Walker, say whatever it was that you wanted to go for. Because look, I am being honest. I really do want Otangelo to be able to feel like he's getting his whole thought out, and I will say that Otangelo sometimes has long thoughts, but that's okay.
Just give me a minute. I'll go to the toilet. Okay.
Well, you're going to be away for a minute.
No, just a minute. Yeah.
Oh, okay, that's fine. Sorry, I thought you wanted another minute to speak, but no, yeah, you can be back in a minute. So the one thing I want to point out is, it frustrates me greatly when Luca is used as an analogy for the first organism, because the first organism was almost certainly orders of magnitude simpler.
Well, I think before even Luca, there comes a point where non-life, like the distinction between life and non-life, is non-existent. Mm-hmm. Right? You're as soon as you have self-replication, you have selection, and as soon as you have selection, you're going to trend towards better and better replication.
Which reminds me.
Yeah.
I have recently, as a result of not having it available, been fed up, and I'm thinking about trying to convince a friend of mine to develop a game together. And does anyone here remember the Creatures game series?
I don't think so.
So Creatures, it started with Creatures, then there was Creatures 2, Creatures 3, there was a kids version, and Creatures 3 plus an expansion is the current, most recent version of it. And so what happened was, you started with these creatures who had both a neural net for decision making and a whole system of biochemistry, both of which were defined by lines of code that when two creatures mated to produce a third creature, the lines of code would be swapped around, and then after the swapping between the two parent lines, there would be random mutations introduced.
Is that Impossible Creatures?
No. This was just Creatures.
Just Creatures. Huh.
And the connections in their neural net were actually enforced by reward punishment systems going on in the simulated biochemistry. And so the idea was you're supposed to get this population of creatures and get them to survive, and based on environmental influences, they'll start to evolve to adapt. And you could watch it happen. And there were various plugins, or in some games, just actually you could just go into the game and look at the actual code governing their genome. And there hasn't been a game like that since Creatures 3, and the servers for the online component, at least, shut down in like 2012 or something. And so I am hoping that Sphincter of Doom and I can work out something. It doesn't have to be super complicated. It doesn't even have to be 3D. But I would like a spiritual successor to that, because I have been missing that degree of artificial life and evolution happening on screen in front of me. Because I used to play a lot of that game, and it was a lot of fun, and you'd see interesting mutations crop up, and there were even mutations that were like completely fatal that never propagated. So every once in a while you would hatch a creature egg, and the resulting creature would just die immediately because there was some fatal mutation. And it was a little sad, but then you drop the corpse into the recycler and get some extra bioenergy, and who cares? Move on.
I posted a wiki link to that game series.
Oh
in
yeah,
case anybody's interested.
yeah. No, that is it. Actually, if you, I'll actually copy it and post it in chat so people can see what we're talking about. If
Came back, guys.
Oh, welcome back.
Hello.
Actually, I guess I can't copy. If someone else who's a mod could do that. We were actually just talking about a biology-based game series that I like, Otangelo, that I would like to see a spiritual successor to, since the rights owners are doing nothing.
You can get it for $6 on GOG.
Yeah, actually, if anyone wants one of the most compelling artificial life sims that's ever been out there, get Creatures 3. I think it's, or there's like some new name for it or whatever. It was the latest version that they released. It's on GOG.
Exodus. They call it the Creatures
Exodus?
3 modernization is apparently called Exodus.
Yeah, okay, yeah, it's Creatures Exodus. I will say one thing though. I highly recommend that you, this is a little bit complicated, but I highly recommend that you edit the starting genome for each creature to give 1/2 of the initial amount of punishment and reward chemicals, because the half-life is too long in the original Creatures 3. And so they end up being rewarded for actions that were taken after the actual action that gave them the initial reward or punishment chemical. And reward punishment chemicals affects the neural net, and it strengthens or weakens connections between the neurons and the neural net. So
Is it basically like selection, or?
well, so what happens? Yeah, there's actually a selection pressure that happens in the neural net. So when a creature takes an action that is, according to its instincts, rewarding, it releases a reward chemical, and that makes that neural net link between the two neurons. Or actually, there's 3 neurons ultimately in any given decision, at least 3. That strengthens all those links, so that they're more likely to be activated the next time the creature is in the same situation.
Has the source code for that entered the public domain? Is there a GitHub?
I don't know that it's in the public domain, although you are allowed to freely publish edited genomes.
Okay.
So, now this was a bigger problem in Creatures 2 than in Creatures 3. In Creatures 3, it's really not necessary, but it can help. Or alternately, you can also reduce the half-life of the reward and punishment chemicals. But that's separate from what we were talking about with Otangelo. So, Otangelo, did you have something you want to say before you headed out? Or no, actually, we're on to Walker. Walker was going to respond to you. So, Walker, go ahead. Sorry about that.
Oh, I don't remember what my specific question was. It was just something. It was something along the lines of like, I don't think. So under the definition of information being that which can be, what? How did you describe it? Something
that which can distinguish one thing from another.
Okay, yeah. So under that definition, like the flow pattern of 2 different rivers is information. So you don't
constrains
Correct.
possibilities.
Yeah.
Basically that. It constrains possibilities.
Okay. Well, I just don't see how that necessarily confirms a designer, right? Because we have, we have examples of information with that definition that we know are not designed, as well as definitions that we know are designed.
Yep. In
Right.
fact,
So you need to either have a definition of information that distinguishes between the two, or, and even then, you would still have to show that DNA fits in the category that includes designer. Right. I don't think you can use just the definition of information or claiming that DNA isn't information as evidence for a designer. If that makes sense.
It's powerful evidence for a designer, because you need hardware and software at the same time, and they have to merge together. And you need also the information transmission systems, and that is also something which is essential. And if you don't have all of this, and you need genetic code as well, that are already 4 different things which have to work in an integrated fashion together in order to have that information transmission system which does things in the cell. But that cannot emerge separately.
Otangelo, in the chat, you asked if I can make you a mod so you can share a paper.
Yes.
I will allow that. I cannot guarantee that I will keep you on as a mod because I'm not currently looking for additional mods. I feel like my mod team right now is sufficiently
No
sized.
problem. Just since I'm here, so someone made a claim, and I would like just to answer.
Yep, you are currently a moderator. I'll be honest, the chances that I remember to change that is low, but
well,
behave.
Don't worry, I will not misuse.
Okay,
my mod.
Don't abuse it for the time that it lasts. No. Actually, also we have another $10 super chat from El Spaghetto who says, "We are listening, Otangelo, but I really love to poke fun. Sorry, I just like that. That's the first thing that happens. Is the difference between Tangelo and Otangelo the same as the difference between possum and opossum?" You don't actually have to answer that, Otangelo, if you don't want to.
That's pretty good.
No, that was amusing, and it wasn't mean. It was just fun. Yeah. So that was.
You have a fun name, Otangelo.
You do actually. I like saying it. Otangelo. It sort of rolls off the tongue after I figured out that there was no C in there, because for a long time I was like, it's Octangelo, right? And I was like, no, it's not. There's no C after the O. What is wrong with me?
Yeah, some guys misspelled my name with a C in the middle.
Yeah.
And some guys they say Otangelo.
Yeah, man. I'll be honest. When I realized that I was doing that, I felt bad for like a day. I was like, oh, because like I really don't like getting people's names. Like, if it's something that's unpronounceable, I'll try. But I'll also be honest and say that I've managed to correctly pronounce Igbo names after being correctly introduced to how they're actually pronounced. And if you know anything about the language Igbo, then you'll know that's not easy. So I try really hard. I used to know a guy whose name was Aglegbe. Yeah, try saying that.
I have one Igbo friend, but his first name is extremely easy to pronounce, so I do
helps.
not have to worry about that. Yeah.
Yeah. So yeah, I try super hard. In fact, I will look up names and get like the International Phonetic Alphabet tight transcription for them, just so I make sure I'm not mispronouncing names.
My mom told me she named me Mike, so I was easy to yell at.
Oh, that's a good reason.
Yep.
Sorry, that's way off topic. But actually, I'll be honest, there is something that we haven't touched on that came up in the debate that I would really like to dig into. And that's just the philosophy of science.
Ooh, that's a good topic.
Because it was something that, what was it, in the last 10 or 15 minutes of the debate, that came up, right? Falsificationism versus confirmationism and things like that. Is that something you'd be interested in discussing, Otangelo?
Yes, I am. To be honest with you, I was a little bit balanced in regards if I'm really using a tautology. So we can go into that, and I'll try to understand that.
Okay, and I just want to answer a quick question from Smitty, who says, speaking of funny-sounding stuff, you still haven't watched Who Killed Captain Alex in Klingon, have you? I have not. Smitty, email me a link to that, because I would be fascinated to watch Who Killed Captain Alex in Klingon. Because, in case you guys don't know, the fact that I really like languages is, and that I'm a gigantic nerd. I mean, I have a Star Trek replicator in my room. It means that I speak a fair bit of Klingon because it's fun. When I'm like, oh, I'm bored, sometimes just for funsies, I study some Klingon. So, please send me a link to that. But okay, so the philosophy of science is something that has actually been argued over for a really long time. Because one of the questions is, science is ultimately, what is it's currently our most reliable method to get rid of false ideas. And ideas that aren't false at least have the possibility of being true. I think we can all agree with that, right?
Yes.
Okay. So, but it's taken a long time to get there. So originally we had, like the natural philosophers in, like, say, ancient Greece, and I don't want to also, like, I need to also include, like, there was natural philosophy going on in, like, ancient China, in Mesopotamia, a bit later, but still happening in places like Mesoamerica and South America. Like, in all of these places, we had people investigating the physical world and trying to come up with explanations that let them make predictions and let them know when to do stuff. Like, when to plant crops, how to build better buildings using less stone or fewer bricks or whatever, right? And then we advanced a bit. And I mean, I'm skipping a whole lot here, but we'll come to like the European Enlightenment, where we were rediscovering, and it's not really fair to say rediscovering, but re-emphasizing is more accurate, a lot of this natural philosophy in Europe. And there was a whole lot of debate about how do we get to the truth. And for a while, what we would now call science was operating under a confirmationism idea. So the idea was, you try to set up an experiment, and if it matches your prediction, you've confirmed your hypothesis, therefore your hypothesis is true, right? But in later times, we realized that confirming the hypothesis isn't necessarily enough. That's actually, it's not even really very useful, because if something meets the prediction, that doesn't necessarily make it true. Because, for instance, let's say I predict, my hypothesis is all female humans are named Lily. That's my hypothesis. I go out and I ask 100 random human females what their name is, and they say Lily. Well, okay, but does that confirm my hypothesis really? Well, no. Because what if tomorrow I find one whose name is, I don't know, any other name? What if I find one who's named Ruth? Well, my hypothesis is now shot down, but I'm not going to do that because I've already confirmed it. That's the problem with confirmationism. That's the reason why now we would say things like science doesn't prove things, it disproves things. So the idea is you need to find some way to disprove a hypothesis, something to falsify a hypothesis. And hypotheses that can't be falsified, those are the ones that we accept, but we only ever accept them provisionally. And eventually, a hypothesis might grow to the point where it is so well supported in the sense that it has gone through so many tests without being refuted, and it explains so much data, and it can be linked up with a number of other hypotheses that have done the same, and together they form a theory. And it's really, really, and I can't emphasize this enough, just extremely important that falsificationism be the foundation of science, because if you just use confirmationism, then you fall prey to confirmation bias, where you only pay attention to the data that you think supports your position.
Okay, can I answer to that?
Yeah, go ahead. Sorry, I'm done.
That is called scientism or verificationism, where someone builds up an epistemological framework based on just science and basis on things that can be tested. And this is a framework which has been given up in the 60s of the last century, and philosophy and theology plays also a relevant part of I would say a correct epistemological framework. And of course, around science there is always a philosophical circle where you make the interpretation of the scientific data.
So
and you have,
okay.
Yeah.
What concerns me about this, what you're saying, Otangelo, is that this isn't. So let's just get out of the way. Scientism is a valid term in philosophy, and it is the belief that the only valid way to knowledge is science. I actually don't know very many people who would accept scientism. Most people would accept that you can gain valid knowledge through purely logical systems like mathematics. Some people would say that personal experience is a valid way to gain knowledge, at least in some areas of life. I actually don't know anyone who, when pressed, would actually go with scientism as their real belief system. I know there are a few people who claim to be empiricists, but I find that they're usually inconsistent on it.
I would consider myself going with scientism.
Would you?
Yeah.
So
interesting conversation.
So you the things that you remember happening, you don't know that any of those things happened?
That what now?
Well, generally speaking, your memories can't be empirically tested with any degree of certainty, with any particularly good statistical levels of certainty, which means
most of your memories Most of your memories are false anyway.
Well, they're false to some degree, but like, I remember my mother's name.
I mean,
can't
I mean,
actually
what-
test it with any statistically significant accuracy.
Maybe I misunderstood your question.
I
I
don't know what
thought you were asking about scientism.
So scientism would generally be the idea that the only way to know something is through science.
No, I wouldn't say that.
Okay.
Well, I think
It's
I
often
think
the best
everything-
way to know some things.
everything can be known with science. Everything.
I actually even disagree there.
Okay.
How
I
would
don't
you
think
use...
you can know anything.
Oh, well, I
that
guess
might be.
Knowledge even possible?
Well, so that's an open question there, but I would say that not many people are strict adherents to scientism.
Yeah, I wouldn't. As far as what I remember, what happened historically, and that has an interesting intersection with trying to figure out abiogenesis and historical science. But I would say that the weight that everything that we need to know about ourselves, the world, everything is all explicable with science. Everything, our minds.
That might be, but that is not strictly speaking scientism.
Not historically what happened.
So, for instance,
Because-
I know that 2 + 2 = 4, but there's not actually any science behind it. That's actually a definitional thing.
I think there is, but
Well, there isn't, because one of the problems that you run into is scientifically how any numbering that you give to objects ultimately ends up being arbitrary, because I could just come up with a different way of numbering them by simply doing things like saying, oh, I don't think that what you identified as a whole thing actually should be identified as a whole thing. So it runs into problems.
so we're talking about two different things, actually.
Well, then we might be. So, but one of the problems is, so I don't think anyone here is saying that science is literally the only way you can be confident about anything. For instance, I have 2 pets. The names that they have are not due to anything to do with science. However, I still know their names. Not that they do, because they're just lizards. They have no idea what's going on. So I don't think that the claim that this is a philosophical naturalism or scientism versus some other epistemological framework is accurate, because the scientific method actually doesn't care a whole lot about your personal epistemological framework. You can have virtually any epistemological framework and still do good science. So I don't see how that's an important aspect of this. And Otangelo, maybe you disagree, and I would like to hear what your response is to the idea that no given person's epistemological framework is terribly important to science. Which, does everyone else agree, generally speaking?
I would, yeah.
I mean, you have to have some extent to which you rely on experience, but otherwise.
But even
if
you write a paper using the scientific method, then your personal epistemology doesn't matter.
Yeah.
Well, right, but I don't know how somebody who didn't rely on experience could operate as a scientist. Is my point. Like you couldn't do the work. You just couldn't do the work.
Yeah, I guess okay. So other than self-defeating epistemologies, how about that?
Right. Yeah.
Okay.
Any reasonable.
Okay.
Like anything that anybody actually holds, to my knowledge.
Yeah. So Otangelo, I don't know if that affects your point in any way, but I would say that I think most people would argue that, with a few exceptions perhaps, because CRISPR did make some good points there, that generally speaking, the personal epistemological stance of any given scientist is not really important to the scientific method or to what degree they follow it.
Now, in our discussions, at least in mine, the relevant issue is always, is there a God or is there no God? That is what I am in dialogue with atheists and non-believers. That is my end goal, to instantiate why I believe that a God is a better explanation for our existence than no God.
Oh, but okay, one of the problems is so I'll bring this up. In the email discussion between you and myself and James, do you remember when I asked James to change the thumbnail so that it no longer said creationist versus atheist?
Yeah, okay, go ahead.
Well, the reason that I do that is because I don't disclose my religious position or lack thereof. Because maybe there is, maybe I don't have a religious position. You don't get to know. But the reason for that is because the kind of conversations I'm interested in aren't about whether or not there's a God. They're about just the science, right? So I'm not here to dissuade you from believing in God. I'm not here to dissuade anyone else from their atheism or to get them to become deists or theists or agnostic or like I only care that you not try to promote ideas that run contrary to science as I mean, I have to go with my own conclusion as to what the consensus in science is and what is the well-supported things. And so those are the ideas that I care about. And so you have to recognize, like, you just have to recognize, Otangelo, it's just a literal fact of the universe, that there are Christian, Muslim, atheist, Buddhist, Baha'i, you name it, people doing science who would disagree with you about things like irreducible complexity or confirmationalism versus falsificationism and things like that, right? Like you get that, right?
Yeah.
So it's great that you want to, or
I mean, a prime example is Ken Miller, which is a Catholic. He is a theist, and he denies irreducible complexity. So
yeah, so
confirms your point.
I don't see how arguing for irreducible complexity, especially given, and I'm just going to be frank here, how completely off-the-wall bonkers nonsense it is, actually helps people become theists. Because the only people that I see being convinced by this argument are either people who are vastly ignorant of biology or people who already have the belief that you're looking to instill.
Okay, my dialogue with atheists is always first to instantiate, is there a God or not? That is the prime question, the first question which I am trying to elucidate with atheists. What comes afterwards, that is religion, that is theology, which God is the true God, that's an entirely different question. But my goal is always about this. I think that the irreducible complexity, of course, if once you acknowledge that there is irreducible complexity in nature, that doesn't lead to necessarily to transcendent God. It can be aliens, it can be farting pixies, it can be whatever you want.
I appreciate that. That was amusing to me. Thank you.
But you're trying to prove, you're basically trying to prove God with science.
No, not proving, Mike. I'm not trying to prove, because God cannot be proven. What I'm using is eliminative inductions and abductive reasoning to the best explanation. So there are basically 2 points which I am making. One is that natural mechanisms, the range of capability to produce what we see in nature, natural mechanisms are not sufficient. That is the negative point. And the second one is positive evidence that certain features which we see in nature, they are positively explained and best explained by an intelligent agent. Like as I always bring up, information, machines, factories, all these kind of things are always being made by intelligence, but not by non-intelligent mechanisms.
You're trying to narrow it down.
Real quick. I just want to read a super chat, because if I don't read them pretty fast, I'll forget. That's just me being honest. El Spaghetto said, let me just take a second to remind Otangelo of the fact that America is having a bigger fish to fry or to fry at the moment. And also, toss some dosh to Dapper. Thank you very much, El Spaghetto. And part of me is really hoping that bigger fish, at least one of them, is catfish, because it has been like a year since I had some fried catfish. And let me tell you, man, I wish I were having fried catfish right now, because I live in
you don't
Phoenix.
Want this fish.
I know what he means, but I'm just taking the opportunity to say, man, I miss fried catfish. The last time I had one was, or last time I had some was last time I was in Alabama, and I went to this particular little like hole in the wall that my friend whose family lives in Alabama recommended to me, and man, I went back there every single day I was down there, and I had so much fried catfish, and so many hush puppies, and yeah. Sorry.
As a native Alabamian, I am actually not a fan of fried catfish. Let the record stand.
What is wrong with you, Walker? Is it because you're a fish?
I do like hush puppies though.
Well, yeah.
I think it's because catfish is garbage fish, but that's
it's okay. Look, for every meat What matters is the preparation.
Oh, I like Southern fried food,
but at the
just
same
not fried
time-
catfish.
At the same time, for every meat, if you deep fry it, it will taste great.
Well,
So
yeah.
that's kind of cheating. Like if you say, like
That's
the
especially
only-
true for gator tail.
The only way you can make this taste great is to do something that would make anything taste great. That's
I feel
pretty
like-
much a confirmation.
I feel like this isn't a problem for me because I still get to go and eat the fried catfish and it's still delicious, and then I eat the hush puppies and they're delicious, and then I use the restaurant's like special homemade hot sauce on both, or well, not both, but
I thought you were
most of
gonna-
them.
Thought you were gonna say use the restaurant's specially made bathroom, because I imagine that's also an issue.
Actually, I didn't really have that issue when I was there. Also, for €5, which that's another thing I wouldn't mind eating is some gyros. And then Zach Brannigan says, "Go try swordfish steak." Oh man, I've had swordfish steak, and I don't want it fried. I want it like grilled, because swordfish is one of those meats that like it's gonna taste good.
Have you ever?
Under a lot of circumstances. Or squid steak. Have you ever had squid steak? From
Oh
Humboldt
yeah,
Squid? Oh man,
funny you said that. I was just about to say actual steak.
Or like
Humboldt
steak. So I so rarely
So
eat
Humboldt's,
steak.
so like most calamari that you have is from market squid, which are pretty small, right?
Mhm.
But also they catch Humboldts, which are like a meter long.
Oh, they're gigantic, yeah.
And from those things, you can get cuts up from the mantle that are huge, that are steak sized.
Are they
And when you
tender?
get some-
Oh yeah,
Yes,
if you cook
when
them
you
right.
get somebody, if you get somebody who knows how to cook one of those things.
Oh my god.
Oh, the taste. The taste is great, right? But what makes it is the texture, because it's the most supple thing you've ever had. It's amazing.
Gotta have one. I gotta have one.
El Spaghetto says for $5. Catfish best fish debate when. Gosh darn, I didn't expect that discussion.
Salmon is the best fish.
So
That's what I had tonight.
salmon is great.
I like it.
Although I will say, if you offer me salmon versus like a nice swordfish steak, that's going to be a hard decision for me.
I would say the best fish is Tiktaalik.
Ooh.
Oh yeah. Oh,
Wait,
I love TikTok.
if we're going phylogenetically, then maybe the best fish is just bacon.
I do not eat pork.
That doesn't make it not
Yeah,
delicious.
you know, there's a lot of things that you got to hide from your extended family down there: catfish, pork, atheism.
Okay, actually, you have no idea how many times my grandma's tried to like sneakily feed me pork and say it's some other meat.
Oh,
no.
Like
that's
I'm not
not
an
okay.
idiot, but there's so many times she'll be like, "Oh no, these ribs are beef ribs," and I'm like, "All right, Nana."
No.
Why
No.
don't you eat pork? Is it because the pigs are intelligent or what?
Yeah.
Yeah.
That's
Pig
mostly-
smart. Yeah.
Because I got some real bad news for you about cows.
Yeah, no, I've been trying to cut out beef as well.
Yeah, I
You ever
guess
see a
yeah,
cow play with a giant ball? It's
All right,
the saddest
it's
thing
so
when you realize
adorable.
that.
Look,
I know,
as
it's-
much as I love this discussion, we can't spend too long on which meat is best. Because
I
as a
just like that a
carnivorous,
cow brain and cow brains are amazingly complicated and like human brains.
Yeah, cows are very intelligent animals, and they're
I suspect
we
they are.
probably shouldn't eat so many of them.
well,
I try to reduce my meat intake, but
I would like to get back to
occasionally. Yes, that's fair.
Otangelo. Sorry about that.
I forgot my point.
So why do you actually? I'll just ask. This is the first question. Do you think that falsificationism is not the correct philosophy under which science should operate? And if so, why?
No, I think in operational science it's okay. I mean, we that's how things in operational science works in order to explain things. In regards of historical sciences, you do not have a time machine to go back and see what happened. So you cannot falsify certain events that happened in the past. So you go with circumstantial evidence.
Okay, so this historical versus operational science is something that was basically made up out of thin air and BS by Ken Ham, and you realize that
Dapper, I
that's
actually need
not
to correct you on that.
fair enough.
That's actually a real distinction. It's philosophers like Carol Cleland. They're actual genuine. They're they're not creationists, but they fully embrace that distinction, but not for the reason creationists say.
Well,
It
Mr.
tends
Wilford,
to be
you have read
more
more.
a
Go ahead.
distinction. It tends to be more a distinction on what they study and the time scale involved. That really seems to be the primary distinction. I have not met many philosophers of science or talked to them who don't believe that ideas about the past can't be tested. But creationists will adamantly say that you can't test an idea about the past. You can only circumstantially look at it. No philosopher of science I've talked to has ever agreed with that. But they agree with the distinction for different reasons.
Okay, so I think that is the real crux of it, is whether or not you can test the past. Otangelo, do you think that we can test the past?
Well, of course, you can make abiogenesis experiments in the lab and see if eventually you put some molecules in the mix and recreate an old atmosphere, what happens, and eventually some amino acids come out, and based on that you can say, oh, okay, maybe that way happened in the past. Okay, that is an experiment.
Well, I think his question is more of, not could something have happened in the past, but did something happen in the past?
Yeah, can we test
So
that?
if you wanted to ask, not could a meteor have struck the Earth 65 million years ago? Well,
66.
Yeah, 66. Okay, I'm sorry. Yeah, it could have, because we can see meteors hit things today, and there's no reason why Earth would be an exception to that. But the question of did it strike the Earth 66 million years ago is a different question.
Yeah, can we test that?
Yeah, when it happens, that is an entirely different question, of course. I mean,
but he's just asking you, could you test the hypothesis that a meteor struck the Earth 66 million years ago?
Oh, no.
Why not?
Well, how do you test it?
Make predictions based on what a meteor strike would do.
Yeah.
One of the predictions that logically follows from a meteor strike is that there would be a crater. Now, that's not a perfect prediction.
Now, you have a creator, you can say
hold up,
happens, but when, that's another question.
Hold up, Otangelo. You interrupted Mr. Wilford a bit, so I want him to get at least a couple more sentences in, and then we will switch to it to you. Is that fair?
Yeah.
Okay.
So, if I have the hypothesis that a meteor the size of Long Island struck the Earth 65 million years ago.
You're breaking up a little bit, Mr. Wilford. Could you get closer to your mic?
Sorry, headphones. Is that better?
Yeah, that's better, yeah.
Okay, so if I have a hypothesis that a 10-mile wide meteor hit the Earth 66 million years ago, there are basic laws of physics that will lead to predictions based off that. And the problem with historical science is when you're testing predictions is you have to make multiple predictions, particularly with fields like my field, geology, because we have a very big problem with evidence degrading over time. So you may successfully predict what an event would produce, but you can't know if a specific piece of evidence would be preserved until the present. So you have to predict multiple things that necessarily would have been made, and then you see whether or not you can find them. If you fail to find one or two, but you find some others, those failed ones aren't falsifications of your hypothesis because evidence will degrade over time. But if you find none of them, that's a problem. Because although we know evidence degrades over time, there are good geologic reasons to reject the idea that all of them would just happen to be erased.
Okay, is that it? Because if it's not, you got to really be fast.
That's it.
Okay. Otangelo, go ahead.
No, I'm not versed in geology, so I have not much to say about that. But another thing which I would like to say is this. Especially in regards of the origin of life, a big problem is the selection problem, which was not there. And you need a specific set of molecules, of building blocks, in order to start life. And in modern cells, these are very complex metabolic pathways which produce these building blocks, which obviously were not there. And that is all about abiogenesis research, trying to find ways how on a prebiotic Earth these molecules could have emerged primarily by non-enzymatic ways, like clay and so forth, and then an eventual transition to metabolic pathways in the cell. The big problem is that there was no selection there. And once you understand that, then you see the huge problem which abiogenesis research faces. You need the right building blocks. Like amino acids, there were hundreds supposedly extant in the prebiotic earth. You have to select 20 which are used in life. The same goes for RNA, which are very complex molecules. And there are very many different sugars down there, which you have to select for. Ribose.
Okay, Otangelo, I'm sorry to interrupt, but that's not addressing the methodology that I was just talking about. Because you can leave geology out of it. It's just the methodology of testing a hypothesis, be it does something happen today or did something happen in the past, is fundamentally the same. You make predictions about necessary consequences of that thing.
That's what I confirmed. That's what I said. You can make tests in the lab, which are being done in the last 70 years. We try to recreate life in the laboratory. And I am just pointing out that one of the big problems is that prebiotically, the right set of building blocks would have to be selected. There was no mechanism to do so.
Hold up. There's two things. One, I'm going to read a quick super chat, and it was for $4.99 from Vandalia 1998, who says, "One of these days you have to have a debate and after show when I'm off." I'm sorry, Vandalia. I didn't even choose this date. So, but the other one is Otangelo. I am not aware of a single laboratory experiment that was attempting to produce from non-living things a completely original event of abiogenesis.
That's not what I'm saying. But
it's literally what you just said.
No. I'm saying RNAs, amino acids, the Urey Miller experiment.
Okay, hold up.
Phospholipids. We are still at that stage.
I want to poll the panel. Panel, is that what you heard, or am I taking crazy pills, Mr. Wilford?
Dude, I'm blanked out right now. I'm sorry, I don't have an answer for you.
Okay, Walker.
Yes.
Okay.
Yes.
Andrew Cumming?
If I said that,
hold up,
Otangelo.
I want to just make sure I'm not taking crazy pills. Andrew, are you still here?
Yeah.
Is that what you heard, that Otangelo said that there were laboratory experiments trying to make new life?
I'm blanking out just like Mr.
Okay,
Wilford.
that's an abstention.
Hey, look.
CRISPR?
I don't know what your medication is, but that's what I heard too.
Okay, Speed?
That's what I heard.
Okay, so Otangelo, maybe you need to rephrase.
Yeah, that's what I said. I mean, the entire venture of abiogenesis research and the ultimate goal is in the end to demonstrate that the life from non-life by no intelligent intervention is somehow possible. That's the end goal. Of course, the experiments, they are not trying to make an entire new cell from scratch, but they are starting with the basic building blocks, and even that is something which they have not achieved even close.
I have 2 points.
That at least is a better phrasing, I think. But one point I want to say is that the goal of science isn't to disprove God. It's to find out how we can explain various phenomena, and because of its very nature, science is restricted to natural explanations. Now, it's possible that, and here's what I would expect. Let's say that the actual explanation for something is, in fact, God had to do it as some kind of miracle involving a suspension of the normal operation of the laws of physics. If that were the case, then what I would expect from science is that we find a hard wall where there's just everything we try doesn't provide any progress at all at a certain point. And what actually we end up seeing is that there is no reasonable non-naturalistic explanation for a particular event. Now, I don't expect abiogenesis to be such a thing, but since it's not currently explained particularly well, I can see that as a possibility. And I want to say real quick, for $2 from El Spaghetto, Otangelo, your sweet accent won't save you now. Actually, that's one thing I want to say about Otangelo's accent. I like the way that you say protein, because it's the exact same way as the kangaroo gym trainer from Agretzko, which is
I got 3 things now. I love the way he says crooks, the crooks of the problem.
Yeah, that's cool too.
Yeah, that's cool too. Anyway, Hubert Yaki believes that he has proved that we'll never find out how life arose. and he is not proving that it could not have arisen. He's only proving that there are so many different ways and possibilities that it could have arisen that we're going to never know for sure. That's one thing. The second thing is that Demeter has told you over and over again about Darwinian selection operating at the molecular basis with very small peptides and RNAs. And you keep missing that, and you keep saying that that's impossible. But you would have to prove that that is impossible, because it is not impossible.
Well, that
Because
is
he has a very
let's let
hypothesis.
Otangelo answer.
Okay.
Well, I've said very many times that, and I have backed up it with many quotes. I have a link just from that. Darwinian evolution starts with DNA replication, and
Nope.
what Demeter claims is that there was somehow a selection prebiotically, and that is where I disagree, because molecules, they do not have the urge to select themselves or even to stabilize somehow to then become information-bearing molecules.
You have
All
never
right, well-
even begun to address Demeter's hypothesis.
I
So-
have addressed it, and he has never answered to me, speech.
So-
You have seen my long replies to all the papers that he has sent to me, and he has not given an adequate reply to that.
Yeah, and in one
So-
of them you said that you didn't read any
I'd
further.
like to
than
get
that,
CRISPR,
right?
CRISPR in here because he's been having something to say for a little bit, and this
So,
is button heads here. So CRISPR, what do you got?
So a strand of RNA that can replicate itself better than other strands of RNA will replicate more because it can replicate better. That is a form of selection. It can happen abiotically if it's capable of replicating itself, which, by the way, will make it replicate really fast. I mean, the idea that you have to have life to have selection is just wrong. And if you think that you do, you're just wrong, and that's the end of it.
Thank you. Thank you.
Selection is
Can
a
I answer to
mathematical
that?
principle. Wait, can I just?
I would like to answer to that.
So I haven't heard from Walker in a little bit, so let's let Walker add on a bit.
Well,
don't
yeah, I'll
take
just
too
keep-
long, and then we'll switch to Otangelo. How's that? Does that work for you, Otangelo?
Okay.
Okay.
I'll keep it. I just wanted to say that selection is a mathematical principle that is a byproduct of differential reproduction, right? So if you have any sort of like favorability of maybe one conformation of a protein in a given solution, you will have selection towards that conformation, just because there's differential success between that conformer and a different conformer.
Okay. So Otangelo, what do you got?
Well, we are far too ahead, because you have to explain how RNAs could have emerged prebiotically, and scientists far off to explain even that. And I have an entire link. I can put it in the site. There are many problems claiming that there was RNA replication. That is just a baseless claim that has not been demonstrated.
They have one pod. They have one pod synthesis for all 4 nucleotides.
Okay, so explain to me then, how was ribose selected amongst all the possible sugars? How was the nucleobases, the pyrimidines and purines, selected amongst many different isomeric compositions which are possible? They used the right composition with carbon and nitrogen to have Watson-Crick base pairing, which is finely tuned. You can put any other atom into the bases like sulfur or whatever other kind of atom you want. How was that selected?
So again, I'm telling you, they've done one pod synthesis. If you want to go through the papers, that's your business, but saying that it can't happen is wrong, because they've put out at least hypothetical models. And by the way, all I need are hypothetical models to disprove your notion that it can't happen. If you say it can't happen, that means I can't come up with a possible mechanism by which it could. I have come up with a possible mechanism by which it could, so you're wrong.
Yeah, you can
Isn't this
also
fun?
say, yeah, you can of course say that fairies, pixies, Santa Claus, whatever, came up with the right composition, and that's it. I mean, if you want to believe that, then I will believe also in Santa Claus.
Well, your idea is pixies to me, so that's what I think of God. So
It's almost like he doesn't understand what a one pod synthesis is. I mean, I don't know what he thinks he's doing in this conversation when he doesn't understand the basics. It's staggering.
Yeah.
So let me ask you: are the scientists making these experiments? How are they in the lab directing the process and everything, or not?
They are starting it and letting it run.
So have they been able to get the nucleobases which are used in life with the right composition of the isomeric composition by just mixing up the atoms and so forth, and they got the right bases? How was that selected? We know even science papers which have found out that there are millions of possibilities, and the ones which are used in life are the best and most fit to do the job. How was that selected if there was no natural
You already
selection?
answered it. The ones that are most fit to do the job are the ones that are going to be used. This is like, you just answered your own question before you finished asking. It's amazing.
No. How did that happen? How does unguided random events?
Because the ones that aren't as good don't get used. That's
good.
how.
So molecules have to go to have something which is good, which works, which makes what's on base, correct base pairing.
Like if they don't do base pairing, they won't replicate properly.
Yes, and why should that the state
because
of
if they can't replicate, because if they can't replicate, because if they can't replicate properly, they won't replicate. And if they can't replicate,
how did it happen prebiotically?
All right, guys, we are coming up on the 2-hour mark, which is where I like to call these. It's also getting very late for everyone. And Otangelo, what time zone are you in? You're in the Eastern time zone, aren't you?
It's almost 3 a.m. here.
Oh, all right. So,
Jesus.
Look, I think it's fair to say that we're probably not going to resolve any of these issues between us, right?
Yeah, this is why I paid in advance.
Uh-oh.
Not gonna do it.
Okay. Well, thank you. It's appreciated. So I want to say thank you to everyone who came by. And Otangelo, I hope that you don't feel like we didn't let you speak your piece, because I have been trying pretty hard to make sure that you'd get to.
No, I appreciate it. I had more time than usually I have, and I've not been so much interrupted. So it was okay. That's why I even stayed so long. You know.
Good. Because I'm not going to say that you're going to have a sympathetic panel here, but I do want to make sure that these discussions can be had in an open environment where people can feel like they can at least get their thoughts out. And that, I think, has happened all around. If you think it hasn't, please let me know, maybe via email, not yelling at me right now.
No. I think it was a nice chat tonight.
Good.
I appreciate it. So.
All right.
I have nothing to complain.
We do have what's probably going to be our final super chat from El Spaghetto, who says, "Everybody have a good night right now," and I second that. If that you celebrate Epiphany, which is the only holiday I'm aware of that's happening right now, happy Epiphany. I hope that your celebration of either, depending on what kind of Christian you are, the baptism of Jesus or the arrival of the Magi to give him his gifts. I hope your celebration of either of those events today was awesome. And if you don't celebrate it, then have a happy January 6th anyway.
Say Dapper, in 9 minutes I will turn 70 years old.
Well, congratulations. Early happy birthday. I'm going to give a little bit of channel news. So this is basically displacing what I would normally have as a Saturday thing. I'm not going to do anything on Saturday after this. So if you're hoping that I was going to have a Saturday thing, sorry, you're getting an extra stream right now. There you go.
Tomorrow at 2, we're going to have the Otangelo show. I call it the weather report, but we're renaming it.
All right.
Well, I'm not guilty that you guys always jump on me when I write something.
You always pop up in chat, no matter what.
you guys.
Awesome. Okay, Otangelo, it's not going to happen tonight, so please remind me to send you those papers, because I do want to send them to you, but I'm done for tonight after this, basically, except for I'm going to hang on this meeting for a little bit because I was requested to answer some questions about fossils, so I will be doing that after we go off air. But, so tomorrow at 4 p.m. Pacific, which is 7 p.m. Eastern. And if you're any of the other time zones, sorry. I'm going to be having a premiere tomorrow. It'll be a fairly long one, so if you're going to want to be here for that, like bring some popcorn and maybe a drink or two or something. I don't know. But it's going to be one of my compilations, because after I finish a long series, I then turn it into a compilation, so you have a video where you can watch the entire pre-scripted series of response videos in a one go without having to hit or watch a playlist or hit, a bunch of links or whatever. That's going to be tomorrow. Again, nothing on Saturday. Tuesday, so a week from yesterday, will be the next Eric with Erica. So if that's a series that you enjoy, that's myself and Gutsa Gibbon, and we respond to craziness that Eric Hovind says. And yeah, that's basically it for channel news. At the end of the month, there may be a disruption in uploads because I am going to be moving, and I don't know to what degree that will cause a disruption. I'm going to hopefully get out at least, or produce at least a few videos so that the Thursday uploads will continue to happen, even if maybe Tuesday, Saturdays are kind of iffy. And with that, I'm going to say we're going to go from my view, from left to right, top to bottom, and get everyone's final words. So, Mr. Wilford, any final words?
It was a good discussion to listen to you both talk, and this whole after show has been very productive, I think. So thank you for having me.
Awesome. All right, Walker.
I thought it was fun, and I should be getting a video out within the next 2 weeks or so. So I'll actually be doing something with my YouTube channel.
Awesome. Andrew Cumming.
Anyone in the audience out there, keep studying, kids.
Nice. I agree. CRISPR.
Thanks for having me on. It was a fun time. Hopefully Otangelo isn't too put out by anything that happened, and otherwise, hopefully we can do it again.
Okay. Speed.
Just plug my channel. We have I'm trying to do a 2 p.m. weather report every day where we get together and discuss what's going on with Standing for Truth, Otangelo, and the others, and try to teach people something. And CRISPR, I would love to have you on sometime. So that's it.
All right. And last but certainly not least, my interlocutor for the last debate, Otangelo. What do you got for us?
Okay, I have 2 channels on YouTube. One is the God Talk, where I make interviews. Next Sunday, we will have Dimitar Kunev, which will have a chat with Sai Garte. And the other channel is Intelligent Design Academy. And I have also a virtual library where I collect the information which I dispose to everyone, which is reasonandscience.catsports.com. So
awesome.
And, if you guys also want to appear sometimes at my channel, you have my email, Dapper, so we can set up something.
Awesome. All right. Well, thank you very much, everyone. I also want to say a special thank you to everyone who super chatted, because super chats really are. Actually, I was looking at my ad revenue and analytics, and super chats are a significant portion of the revenue that this channel generates. And so our super chatters include Sphincter of Doom, Rodent No Last Name, El Spaghetto, Speed of Sound of Gravity, Zach Brannigan, and Vandalia 1998. And I want to say thank you very much to all of you. If you do want to support the channel in other ways, there is a link to my Patreon in the description. There's also a link to my Teespring store in the description. And also, you can join the channel as a member. You'll get access to the Discord server. That's the same server that the patrons get access to, which will allow you to get early access to videos and a few other benefits. And also, if none of that's right for you, please just like and share this video. It really helps out a lot with the algorithm and just broadening the reach of the channel. And as you've seen, I think, I try to have polite and civil discussions about these topics, because dumpster fires are fine, but it's not what I want to do. So thank you very much, everybody, and we're going to get out of here. So goodbye.